Dear listener, hello, and welcome to the Deep Sleep Science Channel. Tonight, we begin with something very small. Not dramatic, not loud, not urgent, just small. Because some of the most remarkable biological systems on Earth begin almost invisibly. A heartbeat you cannot hear. Cells dividing faster than the eye could ever see. A body that has barely formed already following instructions written long before it was born. To understand baby animals is to understand contrast. Immense adults shaped by strength, speed, or scale, and beside them, beginnings that seem impossibly fragile. And yet, nothing about those beginnings is accidental. Every limitation is temporary. Every softness is strategic. Every vulnerability is paired with protection. So tonight, we start with one of the largest bears on Earth. A giant panda can grow to weigh well over 100 kilograms. Its body is built for strength, thick limbs, powerful jaws, dense fur adapted to mountain forests. And yet, when a panda cub first enters the world, it is astonishingly small, often weighing no more than a stick of butter. Pink, nearly hairless, blind. Its proportions barely resemble the adult it will become. The size difference between mother and cub is among the most extreme in placental mammals. At birth, the cub weighs roughly 1/900th of its mother's mass, placing it among the most developmentally immature newborns of any large mammal. Because of this extreme immaturity, the earliest days of a panda's life are defined by warmth and proximity. The cub cannot regulate its own temperature effectively. It cannot crawl with strength. It cannot see. So, survival depends on something quieter: continuous contact, steady nursing, and the precise balance of maternal care. In that extraordinary contrast, immense adult beside fragile beginning, we see something consistent across the animal kingdom. Life does not begin finished. It begins prepared. And tonight, we explore how. Harp seal pups are born on floating pack ice and experience a brief but intense nursing period. For roughly 12 days, they consume milk that is extremely high in fat, allowing them to gain weight rapidly and build a thick, insulating layer of blubber. After this short nursing window, the mother departs, and the pup enters a prolonged fasting period that can last several weeks. During this time, the stored fat becomes the pup's sole energy source, illustrating a compressed strategy of rapid growth, followed by extended independence preparation. Harp seal pups also undergo a visible coat transformation during early development. They are born with a soft white coat known as lanugo, which gives them the well-known white coat appearance on the ice. As weaning approaches, a darker juvenile coat develops underneath, and the white fur is eventually shed. This shift in fur marks not only physical growth, but also transition from maternal dependency toward burly survival stages. White-tailed deer fawns spend much of their earliest days lying motionless in vegetation. Their spotted coats blend with dappled sunlight and forest ground cover, creating effective camouflage. Rather than guarding constantly, the mother typically stays away for long periods to avoid drawing predators. She returns quietly to nurse, relying on the fawn's stillness and concealment as the primary defense system. Although people often say fawns have no scent, this is an oversimplification. Fawns do produce scent, but it is relatively faint in the earliest days, contributing to reduced detectability. Survival depends on a combination of low scent, camouflage, and motionless behavior. Together, these traits form a coordinated strategy where invisibility, rather than speed or strength, defines early life. Mother rabbits nurse their young briefly and infrequently to reduce attention to the nest. Visits often occur at dawn or dusk and can last only a few minutes. This pattern may appear distant to human observers, but it is an anti-predator adaptation. By limiting activity around the nest, the mother reduces scent buildup and visual cues that could attract threats. Baby cottontail rabbits open their eyes within about a week to 10 days after birth. This marks a rapid sensory milestone compared with some other mammals. They also leave the nest relatively early, sometimes by 3 to 4 weeks of age. Their small size and independence often leads people to assume they are orphaned, even when they are developmentally ready to survive alone. Young rabbits disperse before they appear fully grown. Wildlife rehabilitators frequently note that small rabbits encountered alone are often at a normal independence stage. Size alone does not reliably indicate dependence in rabbits. Behavior and physical condition are more accurate indicators of whether intervention is truly needed. Meerkat pups grow up within cooperative social systems where helpers assist with feeding and teaching. Adult group members introduce pups to prey in structured ways. For example, pups may first receive prey that has been disabled or made safer before gradually handling more challenging items. This progression allows skill development in stages. In meerkat groups, teaching is responsive to pup signals. Adults adjust the difficulty of prey depending on the pup's age and begging calls. This feedback loop creates an interactive learning process. Pup vocalizations influence adult behavior, shaping the pace at which hunting competence develops. Polar bear cubs are born in insulated winter dens, extremely small relative to their mothers. At birth, they are blind, sparsely furred, and weigh roughly half a kilogram to a kilogram. Inside the den, protected from Arctic cold, cubs nurse and grow rapidly. By the time they emerge in spring, they are significantly larger and well insulated with fur. Polar bear milk is exceptionally rich in fat, supporting rapid mass gain in a cold environment. This high-energy diet is essential for cubs that must eventually withstand extreme temperatures. The den period allows cubs to convert maternal fat stores into body mass efficiently. Early nutrition directly shapes their readiness to face the Arctic landscape. Giraffe calves enter the world with a dramatic beginning because mothers give birth standing. The newborn drops approximately 2 meters to the ground, a normal part of the birthing process. Despite the fall, calves are adapted for this abrupt start and often stand within an hour. Early mobility is essential in open habitats where predators are present. At birth, giraffe calves can measure roughly 1.5 to 1.8 meters at the shoulder. Even as newborns, they are taller than many adult animals of other species. This initial height supports early movement alongside the herd. However, coordination and steady nursing remain critical during the first days. Echidna young, known as puggles, nurse without nipples. Instead, milk is secreted from specialized pores on the mother's skin. The puggle consumes milk from these patches while sheltered in a pouch-like fold. This monotreme feeding system differs from most mammals but delivers the same essential nutrients. Echidna puggles remain associated with the mother for extended periods. They may stay in the pouch for around 3 months before transitioning to a burrow. Weaning can take many additional months, sometimes up to a year. The extended timeline reflects a slow and steady development strategy. Platypus hatchlings are born blind and hairless after emerging from eggs. Like echidnas, they feed on milk released from pores rather than teats. Milk pools in grooves on the mother's abdomen where the young lap it up. This alternative delivery method illustrates the diversity of mammalian care systems. Platypus's mothers construct burrows that include soil plugs during incubation and early nursing. These plugs may help deter predators and stabilize moisture levels. When the mother pushes past the plug upon return, excess water can be squeezed from her fur. This behavior may contribute to keeping the burrow interior relatively dry. Kangaroo joeys exemplify extreme early birth followed by pouch development. After a short gestation, the tiny newborn climbs into the pouch independently. Inside, the joey attaches to a nipple and continues developing for months. The pouch functions as a protected external nursery. Newborn joeys measure only a few centimeters in length and appear embryonic. They are hairless and have closed eyes at birth. Despite this immaturity, their forelimbs are sufficiently developed to complete the climb to the pouch. This early physical task is essential for survival. Emperor penguin fathers incubate eggs through the Antarctic winter while fasting. The egg rests on the father's feet beneath a brood pouch. By the time chicks hatch, fathers may have fasted for months. The chick's survival is closely linked to paternal endurance during incubation. Penguin chick development varies widely among species. Some, like Adelie penguins, may fledge in a matter of weeks, while others, such as king penguins, can take many months. These timelines reflect differences in climate, foraging distance, and life history strategy. Babyhood length is shaped by ecological context. Some penguin species use a guard phase after hatching. During this time, one parent remains with the chick while the other forages. Parents alternate roles to balance warmth and feeding. This coordinated system ensures the chick remains protected while still receiving food. Bottlenose dolphin milk can be highly energy-dense, with fat content varying over the course of lactation. This rich milk supports rapid growth in aquatic calves. Dolphin calves must swim continuously alongside their mothers. Efficient energy intake helps them keep pace in a three-dimensional environment. Research suggests that dolphin calves may have sensory mechanisms sensitive to fatty acids in milk. Specialized receptors may help detect nutrient content. If calves can sense fat composition, nursing becomes a coordinated interaction rather than passive feeding. Sensory feedback may improve feeding efficiency. Many baby birds develop pin feathers before their adult feathers unfurl. These pin feathers appear as tubular sheaths that later split open. This stage signals progress toward insulation and flight capability. Feather development often proceeds rapidly over days. A fledgling bird is not fully independent, even after leaving the nest. Many species continue to receive food and protection from parents on the ground. This stage can appear awkward, but it represents normal development. Flight skills and foraging abilities continue to improve gradually. Foals are often assessed using early life milestones. They are expected to stand within about an hour, nurse within 2 hours, and pass meconium within 3 hours. These benchmarks serve as health indicators. Delays can signal weakness or underlying medical concerns. Precocial baby animals such as foals and giraffe calves must stand and move quickly due to predation pressure. Early mobility reduces time spent vulnerable at birth sites. This strategy requires advanced coordination at birth. Mothers support the transition through nursing and protective presence. In contrast, altricial babies are born relatively helpless but sheltered. Many songbirds and some mammals fall into this category. Instead of immediate mobility, these young develop within nests or dens, receiving repeated feeding visits until they are capable of independent movement. The harp seal whitecoat stage is defined by both appearance and ecological setting. The white fur provides camouflage against ice and snow. As the pup weans and molts, the coat change coincides with a shift toward aquatic independence. The visible transformation marks a change in lifestyle. Temperature regulation is a central early life challenge for many baby animals. Newborn mammals often rely on body contact and insulated environments. Maintaining warmth supports feeding and immune function. Early life is frequently structured around minimizing heat loss. Camouflage combined with stillness defines survival for many deer fawns. Their instinct to freeze complements their spotted coats. The mother's absence reduces scent concentration near the fawn. Together, these behaviors create a low-detection strategy. Rabbit nests can appear empty even when the mother is caring for her young. Brief nursing visits are easy to miss. Healthy baby rabbits are often quiet and well-fed despite limited adult presence. Observing body condition provides better evidence of care than watching for the mother. Cooperative care in meerkats extends beyond feeding. Helpers shape learning opportunities by adjusting prey difficulty. This group-based approach influences how quickly pups acquire hunting skills. Development becomes a shared investment. Penguin chicks must develop waterproof feathers before entering the ocean. Juvenile down is replaced with feathers capable of repelling water. Until this transition, chicks remain dependent on parents for food and warmth. Feather readiness determines independence. In emperor penguins, successful chick survival depends partly on the father's body condition months earlier. Fat reserves accumulated before winter influence incubation success. The baby's earliest resource is stored adult energy. Environmental conditions long before hatching can affect outcomes. Monotremes such as echidnas and platypuses show that mammalian baby care does not require nipples or live birth. Eggs and milk pores can coexist within the same reproductive system. These alternative strategies expand the definition of mammalian parenting. Baby feeding can occur in multiple anatomical forms. Echidna mothers alternate between carrying the puggle and leaving it in a protected burrow. This flexible system balances foraging needs with baby safety. The burrow provides insulation and concealment, while the mother searches for food. Care involves both contact and strategic separation. Platypus young remain in the burrow for months while nursing. The underground nursery reduces exposure to predators. Much of their early development occurs out of sight. The burrow environment shapes growth and protection. Giraffe calves illustrate the coexistence of strength and vulnerability at birth. Rapid standing ability contrasts with their need for frequent nursing. Following the herd requires coordination and endurance. Early life demands both movement and rest. Harp seal pups demonstrate how brief nursing can produce long-lasting energy stores. Milk is converted into blubber that supports fasting. The pup's early task is energy accumulation rather than hunting. Stored reserves sustain survival during maternal absence. Deer fawns and rabbit kits share the strategy of maternal absence for protection. In both cases, minimal adult presence reduces predator attention. This pattern can appear counterintuitive but reflects an effective survival design. Safety is achieved through concealment and reduced activity. Baby animals are often misidentified as abandoned because human expectations differ from natural strategies. Observers may assume constant parental presence is required. In many species, temporary solitude is normal. True abandonment is better assessed by injury or prolonged distress. Meerkat pups learn to handle dangerous prey through staged exposure. Adults adjust prey condition according to pup age. The structured progression increases competence while minimizing risk. It is one of the clearest examples of teaching behavior in non-human animals. Penguin parenting often requires alternating work shifts. One parent warms the chick while the other searches for food. This division of labor balances energy needs and thermoregulation. Cooperation is essential for chick survival. Many baby animals develop in hidden nurseries such as dens, burrows, or lodges. These structures stabilize temperature and reduce exposure. Hidden early life stages mean much of baby development occurs out of view. The nursery environment is integral to survival. Developmental milestones in baby animals reflect functional thresholds rather than just age. A penguin chick is independent only after waterproofing. A seal pup becomes independent once sufficient boba is stored. Readiness depends on capability, not calendar date. Across species, babyhood is shaped by the first major task each animal must solve. For some, it is standing for others, hiding, swimming, or digesting specialized food. These early tasks define the structure of development. Diversity in baby animals reflects the diversity of ecological challenges they are built to overcome. Across the animal kingdom, baby development often reflects large-scale environmental patterns such as climate, habitat structure, and food availability. Species living in extreme cold, like polar bears or seals, shape early life around insulation and high-fat milk to withstand low temperatures. In contrast, animals living in open grasslands or savannas, such as giraffes and zebras, prioritize early mobility. The scale of the environment, whether frozen sea ice or vast plains, determines the first demands placed on a newborn. Migration routes and ocean currents can influence how seabird chicks grow. In long-ranging species like albatrosses, chick development is tied to parents traveling across immense ocean distances. Because food sources may be far offshore, feeding intervals can be widely spaced. Chick growth therefore aligns with the large geographic patterns that shape adult foraging behavior. In Arctic systems, seasonal light cycles strongly affect baby timing. Emperor penguin chicks hatch during one of the harshest winters on Earth, with adults enduring months of darkness. This synchronization ensures that chicks emerge when adults have completed incubation and can begin foraging again. The scale of the Antarctic winter defines the rhythm of chick development. Desert environments influence the design of camel and llama newborns. In open, hot landscapes, early standing and walking reduce vulnerability to predators and environmental stress. Being able to follow the herd quickly helps desert-born calves remain close to water and shade. Vast terrain shapes the necessity for rapid mobility. Rainforest structure influences primate babyhood over many years. Orangutans, living in complex forest canopies, require prolonged maternal guidance to navigate scattered food sources. The scale of the forest vertical height, seasonal fruiting patterns, and spatial unpredictability creates a long learning curve. The environment stretches babyhood into nearly a decade. Large herd systems affect how foals, calves, and zebra young grow socially. Herd living spreads vigilance across many adults. For babies, this means survival depends not just on their own mother, but on the collective awareness of the group. Social scale becomes part of early safety. Marine mammals operate in three-dimensional space, which changes how babies move and feed. Dolphin calves must coordinate constant swimming while nursing underwater. The scale of open water requires continuous locomotion. Early life unfolds in motion rather than in fixed nests or dens. In ice-based ecosystems, harp seal pups depend on the seasonal stability of pack ice. Their nursing window is synchronized with predictable ice formation and melting cycles. The melting of ice eventually forces pups into the water. Environmental scale dictates the pace at which independence must occur. River systems influence hippo-calf development. Dense waterways create both protection and risk as multiple adults share limited space. Calves must learn to navigate crowded aquatic environments from birth. The scale of the river ecosystem shapes maternal proximity. Mountain habitats affect goat kids and lambs differently than flat plains do. Steep terrain encourages early climbing and jumping. Practicing balance on rocks builds coordination necessary for survival in uneven landscapes. Geography influences motor development. High-altitude environments shape llama and alpaca cria birth timing. Many births occur during daylight, reducing exposure to nighttime cold. Altitude-related temperature fluctuations create selective pressure for early standing and quick nursing. Environmental extremes influence baby timing. Large predators influence how prey species structure babyhood. Fawns, rabbit kits, and cheetah cubs rely on concealment rather than confrontation. In ecosystems where predation risk is constant, stillness becomes a primary early adaptation. The presence of predators shapes baby behavior at scale. Ocean temperature gradients influence whale and dolphin migration with calves. Many species move toward warmer waters for calving. Warmer water reduces energy costs for newborns. Ocean-scale temperature patterns define nursery regions. In cooperative breeders like meerkats, group size affects pup survival. Larger groups can provide more helpers and structured teaching opportunities. Social scale becomes a measurable factor in development. Pup growth is supported not just by a mother, but by an entire social network. Bat maternity colonies represent large-scale communal nurseries. Hundreds or thousands of pups may share a single roost. Colony size influences temperature stability and predator dilution. A large grouping changes the microclimate and protection dynamics. For sea otters, coastal kelp forests provide environmental structure that shapes baby care. Kelp beds serve as natural anchoring systems for floating pups. Habitat complexity becomes part of parenting strategy. The environment itself assists in childcare. In grassland ecosystems, lion cubs transition from hidden dens to pride life. The scale of the pride influences learning and protection. Group introduction marks a shift from secrecy to social exposure. The size of the pride changes the structure of babyhood. Large mammals with extended lifespans, such as elephants, invest heavily in long juvenile periods. Herd size and longevity shape prolonged development. Elephant calves grow within stable family units where multiple adults contribute to safety. Social structure at scale influences calf learning. Arctic dens illustrate how seasonal snowpack influences baby survival. Polar bear cubs rely on insulated dens formed by snow accumulation. Without sufficient snow cover, den quality declines. Large-scale climate conditions directly affect newborn protection. River dam systems built by beavers alter entire ecosystems, and baby beavers grow up inside engineered landscapes. The lodge and dam influence water levels, temperature, and predator access. Habitat engineering shapes baby experience from birth. Seasonal fruit cycles in rainforests influence orangutan infant dependency. Mothers must navigate unpredictable fruiting patterns. Long dependency allows infants to learn spatial maps of resources. Ecological scale dictates learning duration. In temperate forests, raccoon kits develop within dens often located in trees or abandoned burrows. Forest structure provides vertical escape routes and shelter. The scale of woodland habitat frames early exploration. Savanna ecosystems influence zebra foal development. Open visibility requires constant herd movement and early recognition of maternal stripes. Foals must adapt to wide, open landscapes quickly. Space without concealment favors mobility. Marine ice loss can alter harp seal pupping success. Ice stability determines whether mothers can nurse successfully. Large-scale environmental change thus affects baby survival rates. Climate patterns ripple into early life stages. Penguin colonies form massive aggregations during breeding season. Colony density affects chick thermoregulation. Huddling behaviors at scale conserve heat in extreme cold. Group size becomes a thermal resource. Desert predators influence camel-calf vigilance. Wide open spaces mean threats can approach from a distance. Early standing allows calves to remain mobile in exposed environments. Habitat openness drives developmental urgency. Mangrove ecosystems influence crocodile hatchling survival. Nest location relative to water and vegetation affects exposure. Large tidal patterns and seasonal flooding determine nesting success. Environmental cycles shape hatching timing. Sea turtle nesting beaches are selected based on sand temperature and location. Temperature can influence hatchling development and sex ratios. The scale of beach environment directly impacts early survival. Environmental cues shape emergence behavior. In Arctic fox dens, tundra landscape features determine den placement. Elevated areas reduce flooding risk. Geography influences safety from both predators and weather. Baby survival begins with landscape choice. Orangutan populations in fragmented forests may alter infant learning landscapes. Reduced canopy continuity affects movement patterns. Habitat scale shapes what infants must learn. Fragmentation modifies baby experience. Elephant migration corridors influence calf social exposure. Herds moving across large territories expose calves to varied environments. Travel distance broadens learning opportunities. Landscape connectivity affects early exploration. In oceanic systems, whale calves often migrate thousands of kilometers with mothers. Long-distance travel exposes calves to changing temperatures and food distributions. The ocean scale becomes part of baby development. Bat roost selection at landscape scale influences pup growth rates. Cave temperature and humidity vary regionally. Regional climate differences affect maternity colony success. Habitat scale shapes early life physiology. In prairie ecosystems, bison calves must adapt to open grasslands and large herd dynamics. Herd size and migration influence calf positioning and protection. Large group movement shapes early behavior. Forest canopy height influences gorilla infant transport. Dense vegetation requires constant maternal carrying. The three-dimensional structure of habitat defines infant movement strategies. Environmental architecture shapes baby locomotion. Island ecosystems influence seabird chick survival rates. Limited predator presence or absence alters nesting strategies. Geographic isolation shapes babyhood differently than continental systems. Environmental boundaries affect development. In wetlands, hippo calves depend on water depth and seasonal rainfall. River expansion and contraction change nursery space. Hydrological cycles frame calf experience. Coastal erosion can affect sea turtle nesting sites. Shifting shorelines modify hatchling orientation cues. Environmental change at scale influences first journeys. Temperate climates influence deer fawn birth timing. Spring births coincide with vegetation growth. Seasonal cycles ensure milk production matches forage abundance. Climate rhythm shapes neonatal scheduling. Rainfall variability influences antelope calf survival. Drought conditions can reduce maternal condition. Large-scale climate events cascade into early life. Savanna fire cycles influence grazing species birth timing. Post-fire regrowth affects milk quality. Ecological disturbance shapes baby nutrition. Marine upwelling zones influence seal and sea lion pupping colonies. Food availability offshore determines maternal foraging success. Ocean productivity at scale shapes pup growth. In Arctic systems, snow depth affects den insulation for fox and bear cubs. Insufficient snow can expose dens to cold. Seasonal snowpack becomes part of baby shelter. High-altitude oxygen levels influence llama cria physiology. Adaptations to reduced oxygen are present at birth. Elevation scale shapes respiratory development. Tropical rainforest density affects chimpanzee infant travel routes. Branch strength and canopy continuity determine maternal carrying strategies. Habitat structure frames baby movement. Urbanization can influence raccoon kit development. Access to anthropogenic food sources changes learning patterns. Human-altered landscapes reshape babyhood experiences. Coral reef health influences dolphin nursery areas. Prey distribution affects where mothers raise calves. Ecosystem scale guides maternal choices. In polar oceans, sea ice distribution influences seal pupping patterns. Ice flow size determines spacing between mothers. Environmental layout structures early separation. Grassland visibility influences cheetah cub hiding strategies. Vegetation height affects concealment success. Landscape openness drives maternal relocation behavior. River delta complexity influences crocodile nesting sites. Water access and soil composition shape nest success. Geographic features guide hatchling survival. Forest mast cycles influence squirrel reproduction. Abundant nut years can support higher juvenile survival. Resource pulses shape baby population trends. Mountain goat kids are born in steep terrain. Cliff faces reduce predator access, but require early balance. Terrain scale defines motor development. Mangrove root networks provide fish nursery habitats that indirectly support marine mammal mothers. Food webs at scale influence milk availability. Ecosystem productivity shapes baby nourishment. Prairie dog colonies influence pup development through social complexity. Burrow networks provide thermal stability and safety. Colony scale defines early environment. Tidal rhythms influence seal haul-out sites. Timing of tides affects when mothers leave pups. Lunar cycles intersect with baby care. Large river migrations of wildebeest influence cafe survival through sheer numbers. Predator saturation reduces individual risk. Population scale becomes a protective factor. Ocean depth gradients influence whale calving areas. Shallow waters may provide calmer conditions. Bathymetric scale shapes nursery choice. Arctic daylight cycles influence polar bear cub emergence timing. Spring light conditions align with hunting opportunities. Seasonal scale structures den exit. Island nesting sites for seabirds reduce mammalian predator presence. Geographic isolation increases chick survival odds. Landscape separation shapes baby strategies. In tundra systems, lemming population cycles affect predator reproduction. Predator abundance influences risk levels for other newborn species. Ecosystem fluctuations shape babyhood indirectly. Savanna watering holes influence elephant calf social encounters. Shared resources concentrate multiple herds. Environmental focal points expand calf social exposure. Wetland vegetation density influences duckling concealment. Plant structure shapes hiding effectiveness. Habitat complexity affects early vulnerability. Open ocean gyres influence sea turtle hatchling dispersal. Currents transport hatchlings into feeding grounds. Ocean circulation patterns guide early journeys. Forest fragmentation can alter bat maternity colony distribution. Roost availability affects pup clustering. Habitat scale impacts baby growth rates. Desert mite temperatures influence fox kit activity. Cooler evenings allow safe exploration. Climate variation shapes daily baby behavior. Polar ocean prey distribution influences walrus calf growth. Food abundance supports maternal milk production. Large marine ecosystems sustain newborn development. Temperate meadow structure influences rabbit kip survival. Grass height and density determine concealment success. Landscape features frame early hiding. Ocean salinity gradients influence fish nursery zones, indirectly affecting marine mammal mothers. Food web connections ripple into milk supply. Ecosystem chemistry shapes baby nourishment. Coastal cliff availability influences seabird nest placement. Nest height affects predator access and wind exposure. Geological scale shapes chick safety. Grassland herd migration speed influences calf stamina development. Calves must maintain pace over long distances. Movement scale shapes endurance training. Forest understory density influences fawn bedding site choice. Vegetation complexity enhances camouflage. Habitat structure guides maternal decisions. Long before many baby animals take their first breath, their developmental path has already been shaped by millions of years of evolution. The contrast between precocial young, such as foals and giraffe calves, and altricial young, such as songbird nestlings or rabbit kids, reflects different evolutionary solutions to environmental pressures. In open habitats with heavy predation, early mobility evolved as a survival necessity. In more sheltered environments, delayed development allowed energy to be invested into rapid growth and brain expansion within protected spaces. The evolution of mammalian lactation provided newborns with a reliable postnatal nutrient source. Early mammal ancestors developed milk-producing glands that gradually became specialized structures, supporting extended parental care. This innovation allowed young to be born at earlier developmental stages in some lineages, shifting part of growth from inside the womb to the protected environment outside it. Monotremes, such as echidnas and platypuses, represent an ancient branch of mammalian evolution that retains glaying while also producing milk. Their young hatch from eggs but still depend on maternal milk for growth. The absence of nipples in monotremes reflects a distinct evolutionary pathway. Milk secretion through skin pores illustrates that lactation predates the development of teats in mammalian history. Marsupials evolved a reproductive strategy in which extremely underdeveloped young complete much of their growth externally in a pouch. Kangaroo joeys are born after short gestations and continue development attached to a nipple. This strategy likely evolved as an adaptation to fluctuating environments. Mothers can pause or terminate development earlier if conditions deteriorate, reducing reproductive investment during harsh periods. Placental mammals evolved longer gestations supported by a complex placenta. This structure allows extensive nutrient and oxygen exchange between mother and fetus. The result is newborns that may be more developed at birth compared to marsupials. Placental evolution shifted much developmental work to the prenatal stage. In birds, the evolution of flight influenced chick development patterns. Altricial chicks, common among songbirds, hatch helpless but grow rapidly in nests protected from ground predators. Precocial chicks, like those of ducks and shorebirds, evolved in open nesting sites where early mobility increased survival. The divergence reflects ecological trade-offs shaped over evolutionary time. Penguin evolution in cold southern oceans selected for dense down and prolonged parental care. Emperor penguin chicks, in particular, are products of evolutionary pressures imposed by Antarctic winters. Their dependence on brooding behavior and colony huddling reflects adaptations to extreme cold. Each chick carries traits refined by long-term climatic stability and harshness. Seal species that nurse for very short periods represent an evolutionary strategy emphasizing rapid fat accumulation. Harp seal pups, for example, must build blubber quickly in unstable ice environments. This compressed nursing schedule evolved where maternal absence becomes necessary for foraging at sea. Energy-dense milk is the evolutionary solution to limited nursing windows. In large herbivores like giraffes and antelopes, early standing ability evolved as a response to predator-rich landscapes. calves that could rise and move quickly were more likely to survive. Over generations, selection favored musculoskeletal systems capable of supporting weight almost immediately after birth. Developmental timing became synchronized with predation risk. The evolution of camouflage in deer fawns reflects natural selection for concealment. Spotted coats blend with dappled forest light. Forms that remained motionless were less likely to be detected. Behavioral stillness and physical patterning co-evolved as a coordinated defense. In rabbits, the strategy of brief maternal visits likely evolved to reduce scent trails leading to nests. Predators that rely on smell would be less likely to locate young. Selection favored mothers that minimized time at the nest while still delivering nutrient-rich milk. Baby rabbits adapted to thrive with limited direct contact. The evolution of cooperative breeding in species like meerkats allowed multiple adults to contribute to pup survival. This system increases the number of individuals protecting and feeding young. Pups raised in cooperative groups benefit from shared vigilance and teaching behaviors. Social complexity evolved as a survival advantage. Cetaceans evolved live birth in water, requiring specialized birth positioning such as tail-first delivery in whales. This adaptation reduces drowning risk during parturition. Marine mammal calves evolved strong swimming instincts immediately after birth. Selection favored newborns capable of rapid orientation and surface breathing. The extended juvenile periods of elephants reflect the evolution of complex social cognition. Calves must learn social hierarchies, communication signals, and migratory routes. Long lifespans favored investment in prolonged learning. Evolution linked intelligence with extended developmental phases. Orangutans exhibit one of the longest juvenile periods among mammals. Forest fragmentation and seasonal fruit cycles likely shaped this extended dependency. Young orangutans evolved to remain with mothers for many years, learning intricate feeding and nesting behaviors. Cultural transmission became part of evolutionary strategy. In carnivores such as wolves, pack-based feeding of pups through regurgitation evolved as a cooperative solution to provisioning. This allows adults to transport partially digested food efficiently. Pups adapted to digest semi-solid food early. Group-based feeding enhanced survival rates in social hunting species. The evolution of altricial bird chicks may have allowed parents to invest in smaller eggs. Instead of developing fully inside the egg, much growth occurs post-hatching. This shift reduced egg size while maintaining survival through intensive parental feeding. Nest-based care compensated for early vulnerability. Reptiles often evolved minimal parental care strategies, relying on egg number and dispersal. Hatchlings must function independently soon after emergence. This approach differs from mammalian strategies, but reflects evolutionary balance between offspring quantity and parental investment. Sea turtles evolved synchronized hatching behavior, where many hatchlings emerge together. Mass emergence reduces individual predation risk. Selection favored timing coordination to overwhelm predators. Collective movement evolved as a survival mechanism. In crocodilians, some species evolved nest guarding and vocal communication between hatchlings and mothers. This represents an intermediate level of parental care among reptiles. Hatchlings that responded to maternal calls increased survival odds. Evolution shaped acoustic communication before birth. Bats evolved communal maternity colonies where warmth and social grouping enhance pup survival. Large colonies stabilize temperature and reduce predation risk. Pups adapted to cling securely in crowded roosts. Social posturing became an evolutionary thermal strategy. The development of blubber in seal and whale calves reflects adaptation to cold aquatic environments. Fat storage supports thermoregulation and buoyancy. Evolution favored high-fat milk, to accelerate this process. Energy-dense nourishment became essential in marine systems. In primates, grasping reflexes evolved to allow infants to cling to mother's fur. This trait is critical in arboreal environments. Infants capable of secure clinging were less likely to fall. Motor reflexes became embedded in early development. In hoofed mammals, limb elongation at birth reflects selection for speed. Foals and calves must outrun predators soon after birth. Long legs and precocious neuromuscular coordination evolved in response to open landscapes. Birdpin feather development reflects the evolution of feather sheaths that protect growing structures. This design minimizes damage during growth. The staged unfolding of feathers is a product of aerodynamic refinement over millions of years. Milk composition variation among species evolved according to ecological demands. Seal milk is high in fat, while primate milk contains different nutrient balances. Each composition reflects evolutionary tuning to growth rate, environment, and maternal foraging patterns. Camouflage in penguin chicks differs from adult coloration. Juvenile plumage evolved to match colony terrain rather than ocean hunting needs. Color transitions mark shifts in ecological roles. Evolution aligns appearance with stage-specific survival tasks. Burrowing behaviors in foxes and rabbits evolved to create stable microclimates. Underground dens buffer temperature extremes. Young that developed within insulated burrows had higher survival rates. Denning behavior became genetically reinforced. In social insects, complete metamorphosis evolved to separate larval feeding roles from adult reproduction roles. Though not mammals, insect developmental strategies illustrate evolutionary diversification of babyhood functions. Frog metamorphosis evolved as a way to exploit both aquatic and terrestrial niches. Tadpoles feed and grow in water before transforming. This dual-stage life cycle increases ecological flexibility. Evolution partitioned habitat use across life stages. The development of waterproof feathers in penguins evolved alongside aquatic specialization. Down insulation precedes waterproofing. This sequence reflects evolutionary layering of thermal and aquatic adaptations. In whales, long maternal bonds evolved alongside large brain size and social learning. Calves benefit from extended guidance. Cultural transmission of migration routes and vocalizations became part of evolutionary success. The evolution of predator avoidance behaviors, such as freezing in deer fawns, demonstrates behavioral selection. Young that remained still survived more frequently. Over time, instinctive stillness became embedded. In goats and mountain sheep, climbing ability evolved in response to rocky terrain. Kids born with strong coordination had improved escape options. Terrain sculpted musculoskeletal evolution. In bats, wing membrane growth during pup development reflects evolutionary adaptation for powered flight. Wing proportions at birth are small but expand rapidly. Flight readiness is a key evolutionary milestone. Elephant trunk coordination evolves over time, but calves begin practicing from birth. Early trunk experimentation reflects selection for a highly versatile feeding tool. In cheetahs, cub mantles may resemble honey badgers, potentially deterring predators. If resemblance provides protection, natural selection would reinforce this trait. In giraffes, ossicones are present at birth but remain soft. They later fuse to the skull. This delayed hardening prevents injury during birth, reflecting evolutionary compromise. Harp seal coat coloration evolved for snow camouflage. White fur conceals pups against ice. As pups transition to water, darker fur replaces white, reflecting ecological shift. In primates, extended brain growth after birth evolved to allow flexibility in learning. Neural plasticity during infancy supports complex social systems. In cooperative breeders, teaching behaviors may evolve when group structure enhances survival. Meerkat adult adjustments to pup skill level suggest evolutionary roots of responsive teaching. Marine mammals evolved thick skin layers to protect calves from cold water. Thermal insulation remains a core selective pressure in polar oceans. Long lactation periods in elephants evolved alongside large body size and slow life histories. Extended care increases calf survival and knowledge acquisition. In altricial birds, rapid post-hatching growth reflects evolutionary pressure to minimize nest exposure. Chicks that grew quickly reduced vulnerability time. In reptiles with temperature-dependent sex determination, incubation temperature influences hatchling sex ratios. Environmental conditions thus shape population structure across generations. Burrow-plugging behavior in platypuses likely evolved to deter predators and regulate humidity. Such behaviors enhance egg and hatchling survival. The evolution of tail-first birth in cetaceans represents adaptation to aquatic delivery. Birth orientation reduces respiratory risk at first breath. In hoofed mammals, colostrum composition evolved to deliver concentrated antibodies quickly. Passive immunity at birth improves survival in pathogen-rich environments. Flightless bird chicks, such as those of penguins, evolved dense down to compensate for lack of flight-based escape. Thermal survival became a primary driver of feather characteristics. In primates, strong mother-infant bonds evolved in tandem with social complexity. Extended dependency allows transfer of knowledge beyond basic survival skills. Savanna herd structures influenced zebra stripe evolution, potentially affecting recognition and predator confusion. Foal recognition patterns emerged within this ecological framework. In species with seasonal breeding, timing of birth evolved to align with peak resource availability. Newborns entering food-rich periods had higher survival rates. Across evolutionary history, baby animals reflect adaptations that balance vulnerability with preparation. Each newborn carries the imprint of ancestral pressures, shaped by habitat, climate, and social structure. Baby animals develop within carefully structured physical systems that support survival from the very beginning. Dens, burrows, nests, and lodges function as environmental regulators, stabilizing temperature and limiting exposure to wind, rain, and predators. These structures are not random shelters but engineered microenvironments. Their design directly shapes growth rate, energy use, and stress levels in newborns. Temperature regulation is one of the most important systemic processes in early life. Many newborn mammals cannot maintain stable body temperature without assistance. Body contact, nesting material, and enclosure architecture form an integrated thermal system. Warmth supports digestion, immune function, and steady growth. Colostrum plays a critical role in neonatal immunity among many mammals. The first milk contains concentrated antibodies that provide passive immune protection. Because placental antibody transfer varies by species, colostrum consumption within hours of birth is often essential. This system links maternal immune experience to newborn defense. In aquatic mammals, blubber development is both structural and metabolic. Fat layers insulate the body and store energy. Blubber thickness affects buoyancy and thermoregulation simultaneously. The system integrates energy storage with physical protection. Brooding behavior in birds regulates chick temperature through direct contact. Parents adjust posture to distribute warmth evenly across eggs or hatchlings. Feather insulation complements this contact system. The adult body functions as a dynamic thermal regulator. Milk composition varies widely across species due to metabolic system demands. Seal milk, rich in fat, supports rapid insulation buildup. In contrast, primate milk contains more carbohydrates and supports slower growth patterns. Nutrient balance aligns with developmental pace. The pouch in marsupials operates as an external developmental chamber. It protects underdeveloped young while allowing maternal mobility. Within the pouch, temperature and humidity remain relatively stable. The pouch integrates protection with continued maternal activity. Waterproof feather development in penguins represents a staged structural process. Down feathers trap air for insulation but cannot repel water. Juvenile plumage transitions to dense, oil-coated feathers. This change allows the chick to enter aquatic feeding systems. Herd positioning functions as a systemic defense in ungulates. Calves and foals often remain near the center of moving groups. Peripheral adults act as barriers to predators. Social positioning becomes part of a coordinated defense network. In burrowing mammals like rabbits and foxes, soil structure influences den stability. Depth and insulation reduce temperature fluctuation. Airflow within the burrow maintains oxygen exchange. Structural integrity supports sustained nursing periods. Beaver lodges create aquatic buffer zones that regulate water levels. The lodge interior remains dry while surrounded by water. This system reduces predator access and stabilizes microclimate conditions. Construction behavior shapes newborn safety. Giraffe calves rely on musculoskeletal coordination systems immediately after birth. Long limb bones and joint stabilization enable rapid standing. Neuromuscular coordination activates quickly. Structural readiness is critical in predator-rich habitats. Seal fasting periods demonstrate metabolic regulation. Stored blubber sustains pups during maternal absence. Energy conversion processes are tightly regulated to prevent rapid depletion. Metabolic pacing supports survival. Penguin colony density contributes to systemic heat conservation. Huddling behavior reduces individual energy expenditure. Thermal exchange occurs between neighboring bodies. Collective warmth becomes an emergent property of group structure. Bat maternity colonies regulate humidity through clustering. Body heat from many individuals stabilizes cave temperatures. This environmental buffering supports pup wing growth and hydration. Colony scale affects physiological development. In cooperative species, regurgitation feeding systems transport semi-digested food to pups. This reduces chewing demands on young. Digestive systems mature gradually, while nutrition remains accessible. Feeding methods adapt to pup developmental stage. Fawn camouflage depends on patterned fur interacting with filtered forest light. Spot distribution mimics sunlight through leaves. This visual system relies on environmental lighting conditions. Structural coloration complements habitat design. Rabbit nest lining uses maternal fur to insulate kits. The fur layer traps air and retains heat. Material selection improves thermal efficiency. Structural nest composition directly influences kit survival. Platypus burrows include multiple chambers that regulate moisture. Soil plugs limit predator access and water intrusion. The burrow system maintains stable internal conditions. Structural engineering supports egg incubation and hatchling care. Elephant calves develop trunk coordination through repeated muscular practice. The trunk contains thousands of muscle fibers. Neuromuscular integration improves with use. Structural complexity requires extended developmental time. Foals rely on ligament and tendon strength to support early locomotion. Connective tissues must bear weight immediately. Rapid stabilization prevents injury. Structural resilience underlies precocial mobility. Penguin brood pouches regulate egg and chick temperature. Skin contact transfers heat efficiently. This pouch-like fold forms a protective thermal envelope. Structural anatomy ensures incubation success. In dolphins, echolocation systems begin developing early. Cards produce immature clicks and whistles. Neural circuits gradually refine sound production. Sensory systems mature alongside locomotion. Gorilla infants cling to maternal fur using grasp reflexes. Hand and foot structure supports secure attachment. Muscle development strengthens grip. Structural anatomy aligns with arboreal transport. Mountain goat kids rely on hoof structure for traction on rocky terrain. Cloven hooves provide grip. Early practice strengthens joint coordination. Structural adaptation supports climbing. Camel calves lack developed humps at birth because fat storage accumulates later. Energy storage shifts over time. Metabolic processes gradually deposit fat reserves. Structural change reflects energy balance. Penguin chick down forms a dense insulating layer. Each filament traps air close to the skin. This structure minimizes heat loss in icy climates. Feather density supports thermal regulation. Hippo calves can close ears and nostrils underwater. Muscular control prevents water intrusion. This anatomical feature supports underwater nursing. Structural design enables aquatic feeding. Squirrel kits develop climbing ability as their claws harden. Grip strength improves with growth. Structural changes in limb anatomy enhance arboreal movement. Physical adaptation supports exploration. Wolf pups develop deciduous teeth before permanent dentition. Early teeth assist in processing regurgitated food. Dental transition aligns with diet shift. Structural maturation supports independence. Seal whiskers function as tactile sensors detecting water movement. Vibrissae transmit environmental signals. Sensory systems integrate with swimming mechanics. Structural design enhances foraging. Giraffe ossicones begin as cartilage-covered structures. Gradual ossification strengthens them. Delayed hardening prevents injury during birth. Structural timing balances safety and function. Penguin chick digestive systems process regurgitated fish delivered by parents. Enzymatic activity matures with age. Nutrient absorption efficiency increases over time. Internal systems align with dietary complexity. In bagpups, wing membranes stretch gradually as bones lengthen. Membrane elasticity supports flight readiness. Structural expansion must match skeletal growth. Coordinated development ensures aerodynamic function. Deer, fawn heart rates and metabolism adapt to resting concealment. Energy expenditure remains low during stillness. Physiological pacing supports extended hiding. Systemic regulation reduces detection risk. Otter pups rely on dense fur for insulation rather than blubber. Hair traps air at the skin surface. Grooming behavior maintains fur quality. Structural maintenance ensures warmth. Elephant milk contains nutrients that support steady growth over long lactation periods. Composition differs from fast-growing species. Metabolic pacing aligns with slow developmental strategy. Nutritional systems support prolonged juvenile stages. Platypus hatchlings lap milk from abdominal grooves. Fluid collection relies on surface tension. Structural arrangement compensates for lack of nipples. Feeding mechanics adapt to anatomy. Penguin chicks produce vocal calls that allow parents to locate them in crowded colonies. Acoustic recognition systems are refined. Sound frequency and pattern support identification. Sensory communication integrates with colony structure. Cheetah cub mantles consist of longer guard hairs. These hairs break up body outline. Structural fur variation may deter predators. Coat morphology influences early concealment. Fox kits rely on den ventilation systems. Multiple entrances improve airflow. Structural airflow reduces humidity buildup. Den architecture supports respiratory health. Bison calves depend on thick fur for prairie temperature fluctuations. Insulation supports seasonal adaptation. Hair density changes with growth. Structural coat properties support thermal resilience. Sea turtle hatchlings use flipper strokes coordinated with muscular development. Limb structure supports immediate movement. Skeletal strength must match locomotor demands. Structural readiness enables ocean entry. Penguin colonies align nests to reduce wind exposure. Spatial arrangement improves microclimate conditions. Colony geometry affects chick warmth. Structural layout influences survival. Polar bear dens rely on snow density for insulation. Snow traps air and blocks wind. Structural snowpack determines internal temperature stability. Den integrity supports cub growth. Raccoon kits develop dexterous front paws. Digit mobility supports manipulation skills. Muscle control improves with repetition. Structural hand design enhances learning. Elephant calves, large ears, aid in heat regulation. Blood flow through ear tissue dissipates warmth. Thermoregulatory systems function from early life. Structural anatomy supports climate adaptation. In kangaroo pouches, teats swell inside the joey's mouth, anchoring it securely. This prevents detachment during maternal movement. Structural nipple adaptation stabilizes feeding. Anatomical design ensures safe growth. Sea lion pups rely on vocal recognition systems in dense rookeries. Mothers and pups identify each other by unique calls. Acoustic systems prevent misdirected nursing. Structural sound processing supports reunion. Frog tadpoles possess gills that later regress. Structural respiratory transformation accompanies metamorphosis. Lung development replaces gill function. Systemic change supports habitat shift. Hedgehog hoggarts' spines harden shortly after birth. Initially, soft quills reduce maternal injury. Gradual keratinization strengthens defense. Structural timing balances safety and protection. Penguin chicks accumulate fat reserves before fledging. Energy storage supports transition to independent feeding. Metabolic preparation precedes environmental exposure. Structural readiness underlies survival. Across species, baby animal systems operate through integration of anatomy, physiology, and environment. Every newborn body reflects a coordinated design where structure supports function, and early life unfolds within carefully balanced processes. Milk production in mammals is controlled by a finely tuned hormonal interaction between mother and newborn. Prolactin stimulates milk synthesis, while oxytocin triggers milk release in response to suckling. This feedback loop depends on physical contact and stimulation. The more effectively a newborn nurses, the more efficiently the system maintains milk supply. Colostrum contains high concentrations of immunoglobulins that pass through the newborn's intestinal lining shortly after birth. This transfer provides passive immunity during a period when the infant's own immune system is still immature. The permeability of the gut to these antibodies decreases rapidly within the first day or two. Timing is critical, and early nursing directly influences disease resistance. In seal pups, the rapid conversion of milk fat into blubber involves complex lipid metabolism pathways. Enzymes break down milk triglycerides, which are then reassembled and stored in adipose tissue. This storage is not merely for insulation but also for fueling fasting periods. Metabolic efficiency ensures survival when maternal feeding pauses. Dolphin calves coordinate breathing and nursing simultaneously in water. Muscular control of the blowhole must synchronize with feeding behavior. This coordination reflects integrated neural control systems. Respiratory regulation develops alongside feeding mechanics. In marsupial joeys, milk composition changes over time to match developmental stage. Early milk may differ in protein and carbohydrate balance compared to later milk. The mother's mammary glands can produce different milk types simultaneously for joeys at different ages. Lactation adapts dynamically to growth needs. In penguin chicks, digestive enzymes gradually increase as diet shifts from pre-digested regurgitate to whole fish. The stomach lining matures to process larger food items. This enzymatic scaling ensures that feeding transitions occur smoothly. Digestive physiology aligns with dietary complexity. Foals develop coordination through neural maturation of the cerebellum. Balance and fine motor control improve rapidly in the first hours and days. Muscle fibers strengthen through repeated use. Neurological and muscular systems reinforce one another during early movement. In deer forms, the freezing response is mediated by stress hormone regulation. Cortisol levels influence vigilance and immobility. This physiological mechanism complements camouflage. Hormonal balance supports behavioral survival strategies. Otter pups rely on grooming to maintain the air-trapping properties of their fur. Oil secretions from the skin help preserve insulation. If fur becomes waterlogged, heat loss increases. Maintenance behavior directly supports thermoregulation. Elephant calves use tactile communication to maintain proximity to the herd. Trunk touches convey reassurance and direction. Sensory feedback reinforces social bonding. Physical contact supports emotional and spatial awareness. In back pups, wing membrane growth involves rapid cell division in thin connective tissues. Collagen fibers expand as skeletal elements lengthen. Elasticity must be preserved during stretching. Tissue development balances flexibility and strength. Rabbit kits exhibit rapid gastrointestinal adaptation as they begin sampling solid food. Microbial populations in the gut shift accordingly. Digestive flora support fiber breakdown. Internal ecosystems mature alongside dietary change. Wolf pups develop bite inhibition through play. Feedback from siblings shapes pressure control. Neuromuscular circuits adjust based on social responses. Behavioral refinement emerges from repeated interaction. Seal whiskers, known as vibrissae, detect subtle water currents created by prey. Sensory neurons translate vibrations into neural signals. This tactile system integrates with swimming mechanics. Sensory processing enhances foraging efficiency. Penguin chicks regulate body temperature partly through peripheral blood flow adjustments. Vasoconstriction reduces heat loss in extremities. Circulatory control complements insulation. Internal regulation stabilizes energy use. Kangaroo joeys develop lung capacity gradually while inside the pouch. Respiratory tissues mature as oxygen demand increases. Breathing rhythm adapts to growth stage. Pulmonary development supports eventual pouch exit. Giraffe calves strengthen neck muscles early to support head movement and nursing posture. Long cervical vertebrae require muscular support. Coordination between skeletal and muscular systems stabilizes feeding. Growth must maintain structural balance. Hippo calves coordinate buoyancy control while nursing underwater. Lung inflation and limb motion help maintain position. Balance in water depends on subtle adjustments. Hydrodynamic interaction shapes early swimming. In fox kits, olfactory receptors develop rapidly, enhancing scent detection. Early smell recognition supports den orientation. Sensory systems expand environmental awareness. Olfaction guides exploration. Chimpanzee infants refine grip strength through constant clinging. Muscle fiber recruitment increases with use. Motor learning improves through repetition. Physical contact strengthens neural pathways. Sea turtle hatchlings rely on visual cues from moonlight reflecting on water. Photoreceptors guide directional movement. Disruption of light patterns can interfere with orientation. Sensory input determines early navigation. Goat kids refine jumping technique through repeated practice. Joint flexibility improves with movement. Tendon elasticity increases strength. Mechanical adaptation enhances agility. Hedgehog hoggarts begin thermoregulating independently as brown adipose tissue activates. This specialized fat produces heat through metabolism. Non-shivering thermogenesis supports warmth. Metabolic systems mature alongside growth. Raccoon kits develop tactile sensitivity in their front paws, supported by dense nerve endings. Manipulation skills increase gradually. Neural refinement improves object handling. Dexterity expands environmental interaction. Polar bear cubs rely on high-fat milk to fuel rapid tissue growth. Protein supports muscle development, while fat supplies energy. Nutrient partitioning influences body composition. Growth efficiency depends on balanced intake. Penguin chicks strengthen leg muscles before fledging. Standing for longer periods improves endurance. Repeated posture practice conditions skeletal support. Muscular adaptation prepares for aquatic entry. In primates, mirror neurons may contribute to inattention learning in infants. Observing adult actions activates neural circuits. This neural mirroring supports skill acquisition. Social observation accelerates competence. Beaver kits develop incisor growth patterns suited for gnawing wood. Teeth lengthen continuously. Dental wear balances growth. Mechanical chewing strengthens jaw muscles. Back pups refine echolocation calls through auditory feedback. Sound production improves with practice. Neural circuits adjust based on echo return. Acoustic learning enhances navigation. Elephant calves experiment with trunk grip to improve precision. Neural mapping refines motor output. Repeated exploration strengthens coordination. Fine motor control evolves gradually. Seal pups adjust metabolic rate during fasting. Energy expenditure slows to preserve fat stores. Hormonal regulation stabilizes survival. Physiological pacing prevents rapid depletion. Wolf pups' digestive enzymes adapt as diet shifts from milk to meat. Protease activity increases. Nutrient breakdown efficiency improves. Internal systems align with dietary progression. Penguin chick vocal signatures develop unique frequency patterns. Acoustic identity supports parental recognition. Auditory memory reinforces reunion. Sound differentiation ensures targeted feeding. Foals strengthen hooves as keratin hardens. Weight bearing promotes structural integrity. Repeated locomotion stimulates bone density. Skeletal resilience supports independence. Deer fawns' heart rates fluctuate with perceived threat. Autonomic responses prepare for flight if necessary. Physiological flexibility supports survival. Stress response integrates with concealment. Otter pups learn buoyancy control by adjusting lung volume. Controlled breathing stabilizes flotation. Respiratory coordination improves swimming. Internal regulation supports aquatic adaptation. Kangaroo Joe's jaw muscles strengthen gradually as they transition to solid food. Chewing patterns become more complex. Muscle fiber development aligns with diet shift. Structural maturation supports independence. Penguin digestive tracts lengthen relative to body size as chicks grow. Surface area for nutrient absorption increases. Internal scaling supports higher intake. Digestive efficiency rises with age. Cheetah cubs develop coordination between vision and limb movement during play. Neural integration improves targeting. Motor pathways refine timing. Repetition builds hunting competence. Sea lion pups adjust blood oxygen storage capacity through increasing hemoglobin concentration. Circulatory adaptation enhances diving tolerance. Oxygen management improves with growth. Raccoon kit's exploratory behavior stimulates neural development. Sensory-rich environments encourage synaptic growth. Brain plasticity supports learning. Environmental complexity influences cognition. Elephant calves practice rumbling vocalizations. Sound frequency modulation improves gradually. Vocal cord control strengthens. Acoustic communication expands social bonding. In rabbits, gut bacteria transfer from mother to kits during grooming. Microbial colonization supports digestion. Symbiotic relationships form early. Internal ecosystems mature alongside host development. Back pups strengthen pectoral muscles prior to sustained flight. Wing beats increase in frequency with practice. Muscle endurance improves gradually. Structural adaptation enables aerial mobility. Seal pups' whisker sensitivity increases as neural connections mature. Tactile resolution improves detection. Sensory refinement enhances foraging. Neural growth supports environmental awareness. Foals coordinate visual focus with head movement to track herd motion. Neural synchronization improves balance. Sensory motor integration enhances stability. Penguin chicks regulate appetite signals through hormonal pathways. Leptin and ghrelin influence feeding behavior. Metabolic signaling aligns energy intake with growth demands. Internal communication guides nourishment. Giraffe calves adjust gait rhythm as limb length increases. Step frequency and stride length balance efficiency. Biomechanics refine with practice. Musculoskeletal scaling shapes locomotion. Wolf pups develop olfactory discrimination to identify pack members. Scent recognition supports social cohesion. Neural processing sharpens over time. Otter pups' play behavior stimulates endorphin release. Positive reinforcement encourages skill practice. Neurochemical reward systems reinforce learning. Play enhances coordination. Sea turtle hatchlings' flipper strokes become more efficient as muscle fibers strengthen. Repeated propulsion increases endurance. Mechanical refinement supports migration. Elephant calves' temporal lobe development supports memory formation. Neural growth enhances spatial recall. Cognitive expansion supports herd navigation. Across species, baby animals refine physiological and behavioral mechanisms through gradual repetition. Growth emerges from coordinated interactions between hormones, muscles, nerves, and environment, shaping the path toward independence. At the microscopic level, the first moments of life in many mammals depend on rapid cellular signaling. Hormones released during birth trigger cascades of molecular activity that prepare tissues for breathing, digestion, and temperature regulation. Within minutes, gene expression patterns shift inside cells to activate enzymes and structural proteins. These microscopic adjustments support the newborn's transition from a protected internal environment to the outside world. In colostrum, immunoglobulin molecules move through specialized receptors in the newborn's intestinal cells. These receptors temporarily allow large antibody proteins to cross into the bloodstream intact. The intestinal lining soon closes this window of permeability. On a microscopic scale, cellular transport mechanisms determine whether passive immunity is successfully established. Blubber accumulation in seal pups begins with lipid droplets forming inside adipocytes. Fat cells enlarge as triglycerides are stored in tightly packed molecular structures. Enzymes regulate how quickly these lipids are assembled and retained. Microscopic fat storage underlies the visible thickening that insulates the pup. In dolphin calves, muscle fibers increase in diameter through protein synthesis at the cellular level. Myosin and actin filaments multiply within muscle cells. This microscopic growth strengthens contraction force. Swimming efficiency ultimately depends on these molecular assemblies. Inside a marsupial pouch, epithelial cells lining the joey's digestive tract mature gradually. Microvilli on intestinal cells increase surface area for nutrient absorption. These tiny projections enhance contact between nutrients and absorptive membranes. Microscopic architecture drives growth efficiency. Penguin chick feathers begin as clusters of keratin-producing cells in follicles. Each follicle generates barbs and barbules through controlled cell division. The unfolding of a pin feather reflects precise coordination among millions of cells. Structural warmth begins at the molecular scale. In fawn skin, pigment-producing melanocytes distribute melanin granules into developing hair shafts. The spacing of these pigment cells creates spotted patterns. Microscopic pigment distribution results in macroscopic camouflage. Pattern formation originates in cellular organization. Rabbit kit fur growth begins with activation of hair follicle stem cells. Keratinocytes proliferate and push outward to form the hair shaft. Tiny protein chains assemble into durable fibers. Insulation emerges from coordinated cell differentiation. In elephant calves, neural synapses multiply rapidly during early development. Axons extend and form new connections in the brain. Synaptic plasticity at the microscopic level supports learning and memory. Behavioral growth begins with neural branching. Seal whiskers contain specialized nerve endings embedded within follicles. Mechanoreceptors respond to minute water vibrations. Ion channels within these nerve cells open and close in response to mechanical pressure. Sensory perception is rooted in molecular gating. Wolf pup teeth form through interactions between odontoblasts and ameloblasts. These cells secrete dentin and enamel layers. Calcium and phosphate ions crystallize into hydroxyapatite. Tooth hardness arises from mineral deposition. Bat wing membranes are composed of thin layers of skin supported by collagen and elastin fibers. Fibroblasts produce these structural proteins. Elasticity depends on cross-linked protein chains. Molecular tension allows membranes to stretch without tearing. In otter fur, microscopic barbs interlock to trap air pockets. Each hair shaft is coated with oils that repel water. The air retained between hairs reduces thermal conductivity. Insulation is sustained by microscopic surface chemistry. Giraffe calf bone growth occurs at epiphyseal growth plates. Chondrocytes divide and are gradually replaced by ossified tissue. Calcium deposition strengthens bone matrix. Limb elongation depends on cellular proliferation. Kangaroo joey lung tissue expands as alveoli multiply. Type: the second pneumocytes produce surfactant molecules that reduce surface tension. Surfactant prevents alveolar collapse. Gas exchange efficiency improves as microscopic sacs increase in number. Penguin chick metabolic heat production relies on mitochondria within brown adipose tissue. These organelles generate energy by oxidizing fatty acids. Uncoupling proteins allow heat release instead of ATP storage. Cellular respiration directly produces warmth. In raccoon kits, sensory neurons extend dendrites into the skin of the paws. Mechanosensitive proteins detect pressure. Electrical impulses travel along axons to the brain. Fine tactile control originates from cellular excitation. Polar bear cub fur contains hollow hair shafts. Air-filled cores reduce heat transfer. Keratin layers surround microscopic air channels. Insulation arises from structural voids at the cellular level. Penguin vocal recognition depends on specialized hair cells in the inner ear. These cells convert sound vibrations into electrical signals. Ion exchange across membranes initiates neural transmission. Acoustic perception begins with microscopic movement. Chimpanzee infant brain growth involves myelination of nerve fibers. Oligodendrocytes wrap axons in fatty sheaths. Myelin speeds signal conduction. Cognitive processing improves as insulation around neurons increases. Sea turtle hatchlings' orientation systems rely on magnetoceptor cells sensitive to Earth's magnetic field. Iron-containing particles within tissues may respond to magnetic forces. Microscopic alignment guides large-scale navigation. In goat kids, muscle hypertrophy occurs through enlargement of existing fibers rather than formation of new ones. Protein synthesis within myocytes increases fiber thickness. Strength emerges from molecular accumulation. Hedgehog hoblet spines consist of keratinized cells layered in concentric patterns. Cell differentiation produces rigid protective structures. Microscopic layering defines spine hardness. Penguin digestive enzymes such as pepsin are synthesized in gastric cells. Protein chains fold into active shapes. Molecular structure determines digestive efficiency. Seal pup fasting triggers hormonal signaling at the cellular level. Adipocytes release fatty acids into circulation. Mitochondria oxidize these molecules for energy. Survival during fasting depends on intracellular metabolism. Elephant calf trunk coordination involves rapid firing of motor neurons. Neurotransmitters cross synaptic gaps. Microscopic chemical exchange enables precise movement. Bat echolocation refinement depends on synaptic plasticity in auditory cortex neurons. Repeated sound exposure strengthens specific neural pathways. Learning modifies cellular connectivity. In deer forms, stress responses activate adrenal cells that release adrenaline. Hormone molecules bind to receptors on target tissues. Cellular signaling prepares muscles for rapid response. Otterpups maintain fur integrity through sebaceous gland secretion. Lipids coat hair shafts. Molecular hydrophobicity preserves insulation. Kangaroo joey milk contains bioactive peptides that influence growth. Peptide chains bind to cellular receptors. Molecular communication guides tissue development. Penguin chick feather pigmentation arises from melanin granule deposition within keratinocytes. Pigment organelles distribute color evenly. Microscopic granules shape visible plumage. Foal bone density increases as osteoblasts deposit new matrix. Mineral crystals integrate into collagen scaffolding. Skeletal strength develops cell by cell. Wolf pup olfactory receptors bind odor molecules in the nasal epithelium. Chemical interactions trigger electrical impulses. Scent recognition begins with receptor binding. Sea lion pup hemoglobin concentration increases through erythropoiesis in bone marrow. Stem cells differentiate into red blood cells. Oxygen transport capacity expands at the cellular level. Raccoon kit brain growth involves synaptic pruning. Excess neural connections are eliminated to refine circuitry. Efficiency improves through microscopic reduction. Elephant calf skin thickens as keratinocyte layers multiply. Stratified cell layers enhance protection. Structural resilience originates in epidermal proliferation. Seal whisker follicles contain blood-filled sinuses that amplify vibration sensitivity. Fluid movement enhances mechanoreceptor response. Tactile acuity depends on vascular microstructures. Penguin chick appetite hormones circulate at nanomolar concentrations. Receptor binding alters neuronal firing patterns. Molecular signaling regulates feeding behavior. Cheetah cub visual acuity improves as photoreceptor cells mature. Rods and cones increase in sensitivity. Cellular development sharpens perception. Sea turtle flipper muscle fibers increase mitochondrial density. Enhanced oxidative capacity supports sustained swimming. Cellular respiration underpins endurance. Goat kid tendon strength increases through collagen cross-linking. Chemical bonds reinforce fiber networks. Microscopic alignment determines tensile strength. Harp seal pop blubber cells enlarge through lipid droplet fusion. Intracellular storage compartments merge. Cellular volume expansion thickens insulation. Bat pup wing bones elongate as osteoclasts and osteoblasts remodel tissue. Bone resorption and deposition remain balanced. Microscopic remodeling shapes flight readiness. Elephant calf memory formation depends on hippocampal neuron growth. Dendritic spines multiply to form new synapses. Learning is encoded at the cellular interface. Rabbit kit intestinal cells express lactase enzymes for milk digestion. Enzyme activity declines as solid food intake increases. Gene expression shifts support dietary change. Penguin chick down fibers grow through keratin gene activation. Protein synthesis produces filamentous strands. Feather softness begins in cellular transcription. Wolf pup immune cells proliferate after exposure to environmental microbes. White blood cell populations expand. Immune competence develops through cellular training. Otterpup whisker nerves transmit impulses via sodium channel activation. Ion flux across membranes initiates action potentials. Sensory perception arises from electrochemical gradients. Sea lion pup muscle fibers switch from neonatal to adult isoforms of myosin. Protein composition changes improve contraction strength. Molecular transitions enhance locomotion. Giraffe calf cartilage transforms into bone through endochondral ossification. Chondrocytes calcify before replacement by osteocytes. Limb elongation follows cellular sequence. Penguin chick thermoregulation relies on ATP production in mitochondria. Energy conversion efficiency influences heat output. Cellular respiration sustains warmth. Across all species, microscopic processes govern visible growth. Every movement, call, and milestone in baby animals is built upon cellular chemistry, molecular signaling, and coordinated tissue development, but quietly unfolds beneath the surface. Newborn animals often begin life with senses that are still incomplete, yet already quietly adapting to the world around them. In many mammals, the eyes are closed at birth, and the optic nerve pathways are still refining their connections. As light gradually reaches the retina for the first time, neural circuits strengthen through repeated exposure. Vision does not simply switch on, it stabilizes through steady sensory input and gentle experience. In wolf pups, the ear canals open days after birth, allowing sound waves to reach the developing auditory system. Before this moment, communication occurs primarily through touch and scent. When hearing begins, the brain starts mapping frequencies and rhythms. The early sounds of the den become the foundation for pack communication later in life. Dolphin calves are born with functional hearing in water, an essential adaptation in an aquatic environment. Sound travels faster and farther underwater than in air, shaping how calves perceive their surroundings. Acoustic bonding between mother and calf begins almost immediately. Signature whistles allow calves to recognize and remain oriented toward their mother in a vast ocean. Penguin chicks rely heavily on vocal identification in crowded colonies. Each chick develops a unique call pattern that parents learn to recognize. Amid thousands of voices, subtle variations in pitch and rhythm allow precise identification. Auditory discrimination becomes a survival tool in dense social environments. Elephant calves are sensitive to low-frequency rumbles that travel through the ground. Vibrations can be detected through the feet as well as the ears. This dual sensory system allows calves to perceive distant herd movements. Communication is not only heard but also felt. Sea turtle hatchlings depend on visual contrast between dark land and brighter ocean horizons. Their eyes are tuned to detect the relative light gradient at night. Even subtle artificial lighting can disrupt this orientation system. Visual cues guide the first journey toward water. Backpups develop echolocation gradually, first producing immature calls that lack adult precision. The auditory cortex adapts to interpret returning echoes. With repetition, sound production and perception refine together. Hearing and vocalization evolve in coordination. Giraffe calves rely on sight to track their mothers within open landscapes. Their large eyes provide a wide field of vision even at a young age. Visual tracking supports herd cohesion. Early visual acuity strengthens with exposure to moving forms. Otter pups are initially hesitant swimmers, but tactile feedback from water helps refine motor control. Vibrissae detect subtle current changes around their faces. Sensory information guides adjustments in body posture. Touch and motion integrate to improve coordination. Kangaroo joeys inside the pouch experience limited visual stimulation early on. Gradual exposure begins as they peek outside. Light, movement, and sound increase slowly in intensity. Sensory systems expand as physical development progresses. Fox kits begin life with closed eyes and rely primarily on scent. The mother's odor provides orientation within the den. As vision develops, sensory balance shifts. Smell remains important, but visual exploration begins to dominate. Seal pups respond to maternal vocalizations even before strong visual recognition develops. Sound carries effectively across ice surfaces. Auditory cues support reunion after brief separations. Hearing becomes a guiding signal in expansive, bright environments. Chimpanzee infants rely heavily on tactile contact with their mothers. Constant clinging provides sensory reassurance and warmth. Touch supports emotional regulation and neural growth. Physical proximity shapes early psychological stability. Rabbit kits begin with limited hearing and vision, yet their whiskers provide tactile awareness. These fine hairs detect air movement and surface contact. Gradual sensory opening occurs over days. Perception broadens as neural circuits mature. Hedgehog hoglets initially depend on smell and touch, as their eyes remain closed for several days. The nest environment provides familiar sensory signals. As eyes open, spatial awareness increases. Sensory layering deepens environmental understanding. Penguin chicks can detect subtle changes in parental posture through visual cues. Body language communicates readiness to feed or move. Even simple gestures convey meaning. Observation becomes part of early learning. Sea lion pups use vocal calls and scent recognition to reunite with their mothers in crowded rookeries. Each pair forms a unique acoustic bond. Olfactory cues reinforce this connection. Multiple sensory channels ensure accurate identification. Wolf pups explore their environment with both nose and mouth. Oral exploration supplements scent perception. Taste receptors contribute to environmental mapping. Sensory curiosity supports learning. Elephant calves show early responsiveness to visual gestures from older herd members. Movement patterns signal direction and safety. Imitation may follow observation. Social cues shape coordinated travel. Bat pups refine wing control through proprioception, the internal sense of body position. Stretch receptors in muscles inform balance. Sensory feedback allows fine adjustments during early flight attempts. Internal awareness guides external motion. Deer fawns rely on stillness combined with acute hearing. Subtle rustling sounds may signal approaching danger. Auditory vigilance complements camouflage. Senses compensate for physical vulnerability. Otter pups respond playfully to tactile stimulation from siblings. Roof and tumble interactions stimulate neural pathways. Play reinforces sensory integration. Gentle contact strengthens coordination. Kangaroo joeys gradually adapt to external temperature shifts as they exit the pouch more frequently. Skin receptors respond to airflow changes. Thermal sensation informs behavioral choices. Sensory adaptation supports independence. Penguin chicks exhibit strong responses to hunger signals, guided by internal hormonal feedback and external parental cues. Physiological hunger interacts with social signaling. Sensory systems integrate internal and external information. Cheetah cubs develop binocular vision for depth perception during play. Pouncing accuracy improves with repeated targeting. Visual refinement enhances hunting skills. Perception and movement synchronize over time. Sea turtle hatchlings are sensitive to the slope of the beach beneath their bodies. Tactile feedback helps maintain forward movement. Gravity and ground texture guide direction. Tooch complements vision during the first crawl. Raccoon kits use their highly sensitive front paws to explore textures and objects. Tactile curiosity drives learning. Fine sensory resolution supports problem-solving abilities. Exploration builds neural connections. Elephant calves learn vocal mimicry by listening closely to adult rumblings. Sound imitation strengthens social bonds. Auditory memory supports group identity. Repetition shapes vocal patterns. Seal pups recognize maternal scent even after short separations. Olfactory memory anchors reunion. Chemical signals persist in the environment. Smell reinforces attachment. Foals quickly develop visual recognition of herd members. Pattern and movement cues differentiate individuals. Sight strengthens social awareness. Recognition reduces stress during movement. Penguin chicks respond to subtle wind shifts and adjust posture accordingly. Sensory awareness supports energy conservation. Environmental feedback shapes body positioning. Sensory input guides thermoregulation. Wolf pups gradually distinguish between pack member scents and unfamiliar odors. Olfactory discrimination refines social boundaries. Scent mapping supports territory awareness. Smell underpins social cohesion. Otter pups learn to interpret water sounds, such as splashes and currents. Auditory cues inform diving attempts. Listening complements visual scanning. Multisensory integration improves coordination. Kangaroo joeys experience vibration cues from maternal movement inside the pouch. Rhythmic motion provides sensory stability. Internal sensation contributes to emotional calmness. Early life unfolds within a consistent sensory envelope. Penguin chicks adjust vocal intensity depending on distance from parents. Volume modulation improves communication. Auditory feedback supports effective calling. Sound dynamics enhance survival. Sea lion pups respond to touch from their mothers with immediate calming behaviors. Physical reassurance regulates stress responses. Sensory comfort shapes emotional development. Touch remains foundational. Giraffe calves refine spatial awareness as they navigate tall grasses. Visual and tactile cues interact. Environmental complexity enhances perception. Awareness strengthens mobility. Across species, sensory systems mature in stages, each layer building upon the previous one. From the first flicker of light in a newborn's eyes to the subtle vibration felt through the ground, baby animals gradually assemble a complete picture of their world through touch, sound, sight, smell, and movement. In many large mammals, babyhood unfolds slowly across years rather than weeks. Elephant calves, for example, remain closely associated with their mothers and herd for extended periods, gradually absorbing social knowledge through quiet observation. This long developmental window allows subtle skills to accumulate over time. Navigation routes, water locations, and social hierarchies are learned through repeated exposure rather than sudden instruction. Orangutan infants experience one of the longest dependency periods among mammals. For many years, they remain within arm's reach of their mothers in the forest canopy. Seasonal fruiting cycles teach patience and memory. Over time, young orangutans internalize complex feeding maps that cannot be learned quickly. Whale calves migrate alongside their mothers over vast ocean distances. These journeys span thousands of kilometers and repeat annually. The repetition imprints migratory timing and route familiarity. Knowledge transfers gradually, season after season. Seal pups that fast after weaning rely on stored blubber to bridge a quiet developmental gap. During this time, they remain largely inactive. Slow metabolic pacing preserves energy. Physiological endurance grows silently before first independent foraging attempts. Penguin chicks accumulate fat reserves before fledging, remaining land-bound for months in some species. Their growth appears steady and deliberate. Only once waterproof feathers are complete does the next stage begin. Development proceeds in measured transitions rather than abrupt leaps. Giraffe calves refine gait patterns as their limbs lengthen. Stride adjustments occur incrementally over weeks. Muscle memory builds through countless small steps. Locomotion becomes smoother through repetition. Wolf pups spend weeks learning social boundaries within the pack. Hierarchy recognition develops gradually through play and subtle correction. Dominance and submission cues are not instantly understood. Social fluency forms slowly over time. Otter pups' perfect diving technique through extended play sessions. Early dives are shallow and brief. Depth and duration increase with practice. Confidence emerges through repeated exposure to water. Kangaroo joeys shift from constant pouch residency to brief external excursions. Each outing lengthens slightly. The transition unfolds in small increments. Independence grows quietly within a protective framework. Elephant calves gradually strengthen memory through repeated herd travel. Familiar waterholes and paths become embedded knowledge. Experience layers upon experience. Spatial awareness deepens across seasons. Chimpanzee infants learn tool use through prolonged observation. Years may pass before mastery. Skills mature with patient imitation. Knowledge transfer unfolds through steady practice. Sea lion pups refine swimming stamina over months. Early attempts tire them quickly. Endurance builds progressively. Muscle strength increases through repeated effort. Foals move from unsteady standing to confident trotting within days, yet full coordination takes longer. Balance improves through gradual neuromuscular tuning. Each stride reinforces control. Maturity arrives through incremental refinement. Harp seal pups transition from land-bound rest to ocean immersion in stages. Initial swimming efforts remain near ice edges. Confidence in water develops steadily. The shift from ice to sea unfolds across days. Fox kits expand exploration range gradually beyond the den entrance. Initial ventures are brief and cautious. distance increases over time. Curiosity grows alongside coordination. Bat pups strengthen wing strokes through repeated flapping practice within roosts. Early flights remain short. Muscle endurance builds progressively. Flight competence emerges over weeks. Raccoon kids develop problem-solving skills by manipulating objects repeatedly. Dexterity improves with trial and error. Learning occurs through quiet persistence. Motor skill mastery takes time. Penguin chicks wait for favorable weather windows before first ocean entry. Environmental timing influences transition. Patience becomes part of development. Readiness aligns with seasonal rhythm. Elephant calves experiment with trunk coordination daily. Precision handling improves through repetition. Fine motor control strengthens gradually. Practice shapes capability. Wolf pups expand territory awareness slowly as they accompany pack movements. Early travel distances remain short. Exploration radius widens incrementally. Confidence follows familiarity. Otter pups increase diving duration over weeks. Each dive extends slightly deeper or longer. Endurance builds in measured steps. Mastery emerges through consistency. Sea turtle hatchlings, once in the ocean, drift in currents for extended juvenile phases before returning to coastal waters years later. This prolonged pelagic stage unfolds quietly beyond human view. Growth continues slowly across open seas. Giraffe calves or cicones gradually ossify over time. Cartilage transitions to bone through steady mineral deposition. Structural maturity does not occur overnight. Development advances gradually. Seal pups' fasting metabolism slows progressively rather than abruptly. Hormonal adjustments fine-tune energy usage. The body conserves resources with measured control. Survival depends on steady pacing. Chimpanzee infants refine social play behaviors over years. Roughhousing becomes more nuanced. Subtle communication skills emerge slowly. Social literacy deepens through experience. Kangaroo joeys transition to full independence only after months of partial reliance. Nursing frequency decreases gradually. The final separation feels almost seamless. Independence evolves without sudden break. Elephant calves internalize herd vocalizations over time. Subtle variations become recognizable. Acoustic memory builds quietly. Familiar sounds anchor belonging. Fox kits shift diet gradually from milk to solid prey. Jaw strength and digestive capacity increase incrementally. Each feeding stage prepares for the next. Growth follows a natural rhythm. Penguin chicks molt down feathers slowly as waterproof plumage grows underneath. The change appears subtle, day by day. Transformation proceeds in stages. Visual shifts reflect deeper structural readiness. Wolf pups refine scent discrimination through extended exposure to pack and territory odors. Olfactory maps expand with repetition. Recognition strengthens over time. Sea lion pups build diving tolerance through repeated oxygen management. Breath hold duration lengthens slowly. Cardiovascular adaptation develops steadily. Physiological change mirrors behavioral practice. Foals mature socially through quiet observation of herd interactions. Early curiosity evolves into understanding. Subtle cues shape behavior gradually. Social awareness deepens over months. Otter pups' playful wrestling strengthens muscles slowly. Strength increases through daily repetition. Coordination becomes fluid. Growth unfolds through consistent activity. Elephant calves' tusks begin as small protrusions and elongate slowly over years. Dental structures mature gradually. Visible growth reflects long-term development. Bat pups improve echolocation precision over extended practice. Echo interpretation becomes clearer. Acoustic refinement emerges through repetition. Neural tuning sharpens gradually. Seal pups gradually shift from maternal dependency to independent hunting. Early attempts remain cautious. Confidence in prey capture grows over time. Mastery unfolds patiently. Penguin chicks wait months before first swimming, even when physically capable. Environmental conditions dictate timing. Development respects seasonal cues. Transition aligns with ecosystem rhythm. Chimpanzee juveniles continue learning tool complexity long after infancy. Knowledge layers accumulate slowly. Cognitive growth extends across childhood. Across species, baby animals grow through subtle, steady changes rather than dramatic shifts. Milestones may appear sudden, yet they rest upon countless small adjustments unfolding quietly over time, shaping resilience through patience and repetition. As baby animals move toward independence, many of their earliest survival behaviors gradually soften into routine. What once required intense vigilance becomes familiar habit, woven quietly into daily life. The urgency of newborn vulnerability slowly gives way to steady confidence. Growth often feels less like a leap and more like a gentle unfolding. Elephant calves that once stayed pressed close to their mothers begin walking with relaxed spacing. The tight circle of protection expands, though it never fully disappears. Trust in the herd replaces constant dependence. Security becomes an internalized sense rather than a physical position. Penguin chicks that once relied entirely on brooding warmth begin standing comfortably in the wind. Feathers that started as soft down now provide insulation on their own. The colony still surrounds them, yet their bodies hold heat independently. Readiness arrives through quiet preparation. Wolf pups that once tumbled clumsily at the den entrance start moving confidently across open terrain. The playful boundaries of early life widen into real exploration. Social rules land in play, become instinctive responses. Structure replaces uncertainty without fanfare. Otter pups that first floated hesitantly now dive beneath the surface with ease. The water that once felt uncertain becomes familiar territory. Each movement is smoother than the last. Confidence grows in steady increments. Kangaroo joeys that once spent nearly every moment inside the pouch begin resting outside beside their mothers. The pouch remains available, yet less necessary. Independence forms through proximity, not separation. Growth occurs while safety remains nearby. Seal pups that conserved energy in stillness gradually begin testing the sea. The quiet fasting period gives way to motion. Stored reserves support this transition. What was preparation becomes action. Foals that once wobbled on unsteady legs begin matching the herd's rhythm. Their strides lengthen and settle into a natural cadence. Balance no longer requires visible effort. Strength becomes part of normal movement. Chimpanzee juveniles who once watched intently from their mother's sides start experimenting with tools on their own. Attempts may still be imperfect. Observation gives way to practice. Learning shifts from imitation to experimentation. Giraffe calves, once dwarfed by surrounding adults, grow into their height gradually. The landscape that once towered above them begins to feel proportionate. Perspective changes with growth. The world remains vast, but it feels navigable. Sea turtle hatchlings that began as tiny silhouettes crossing sand drift far out at sea, growing slowly in open currents. Their journey is quiet and largely unseen. Maturity unfolds across years of gradual ocean travel. Bat pups that once clung motionless to roost walls begin hanging independently between fights. The first awkward attempts fade into routine. Muscle memory stabilizes motion. Flight becomes effortless with repetition. Raccoon kits that once investigated objects cautiously begin manipulating their environment confidently. Curiosity deepens into capability. Skill grows without announcement. Familiarity shapes competence. Elephant calves slowly memorize seasonal water routes. Paths once unfamiliar become ingrained knowledge. Memory accumulates gently. Experience builds resilience. Penguin juveniles that once huddled tightly with siblings now stand with measured spacing. Individual strength complements colony support. Community remains, but reliance softens. Balance replaces fragility. Fox kits expand their territory beyond the den until the entrance feels less central. The world beyond becomes ordinary rather than unknown. Exploration shifts from novelty to habit. Growth blends into daily life. Sea lion pups that once depended entirely on maternal guidance begin swimming alongside others. Social interactions broaden. Independence develops within group rhythm. Learning continues quietly. Wolf juveniles gradually contribute to pack hunts, first in small roles. Participation increases subtly. Responsibility grows through involvement. Competence arrives through shared effort. Otter juveniles refine diving without visible strain. Movements that once required concentration become fluid. Skill becomes instinctive. Repetition smooths effort into ease. Kangaroo young that once returned frequently to nurse begin spacing those visits further apart. Dependency fades gradually. Separation is gentle, not abrupt. Maturity evolves in small steps. Seal pups that first enter the water cautiously now move through it with steady control. Buoyancy and balance feel natural. Preparation gives way to proficiency. Confidence replaces hesitation. Foals integrate fully into herd movement patterns. Their presence blends seamlessly into group flow. Individual growth merges with collective rhythm. Stability emerges quietly. Chimpanzee juveniles begin teaching younger individuals simple behaviors, repeating what they once learned. Knowledge circulates without ceremony. Learning becomes part of community continuity. Elephant calves that once needed constant reassurance now offer it to younger herd members. Touch becomes reciprocal. Growth transforms dependence into contribution. Development completes its quiet arc. Across species, babyhood does not vanish suddenly. It gradually transforms into capability, memory, and rhythm. What began as fragility becomes familiarity. And in that gentle progression from need to knowing, life continues steady, patient, and quietly resilient.